slightly green color, but it's not as bright as the tumor itself. Now, however, what I told you is to make muscle, starting with myoblasts, and you fuse together thousands of these myoblasts, and then you make a muscle, okay? And we put the GFP on in every myoblast. So we expected to see the whole muscle being green, but it isn't, just the tumor is green. And the question is why? So we looked very early when muscle is just forming. So in a mouse, this is at what we call 11 days and a half after birth. The whole birth cycle of a mouse is about 19 days instead of 9 months, much quicker. So here is the muscle just forming, and here are the green myoblasts. So you can see that these cells are not participating in making the muscle. And then the question is why? And the reason, turns out, is that they're dying. As soon as the fusion gene is on, those cells start to die. And they're dying by what we call apoptosis. They're dying very, very rapidly, all over, okay, except wherever there's cartilage. So this is a rib, and you can see green cells are still surviving, and the red cells are the ones that are dying, and those cells along the cartilage are still surviving. So what it looks like is the cartilage is producing a factor that allows these cells to survive, then continue to duplicate, accumulate the secondary events, next events that allow that tumor progression. So we're interested to see what are those things that are being made by the cartilage that allows these tumor cells to survive and continue to divide. And we're looking for that factor. I'll take you through this again. The other thing that was surprising is I told you that the cells with the fusion gene are dying. And so what we expected is that all the muscle is dying, and what we expect then is that the mouse shouldn't be able to run around. Every mouse develops tumors, but they're still running around. So that tells us the muscle's still there. And so that was a paradox. But I also told you that there were 2 genes. One is called MIF5, the other is called MyoD, that are present in myoblasts. So if you inactivate one gene, the mouse is still happy. You inactivate the other gene, the mouse is still happy. You inactivate both genes, and now the mouse no longer has muscle, and they quickly die. So that tells us there are 2 ways of what we call redundancy. You have 2 genes doing the same thing, knock one out, the other one's still okay. But there's 2 ways of having redundancy. You could have both genes present in every cell. Alternatively, you could have 2 independent cells, one having one and having the other. Okay? So then if we knock one out, then the other cells could still take over. And that's what we found out. And we could do that simply by doing the same trick I just showed you. In that locus, we have a stop sign flanked by LOXP, and now we put here a really deadly gene, diphtheria toxin. And a diphtheria toxin is so deadly that one molecule can kill a gene, and kill a cell. And so we have that gene present, and now if we now put that in the presence of MIF5, that is what's driving our creed, we kill all the cells that those myoblasts. And it turns out that those mice are okay. The other gene, the other cell, has MyoD. That cell population expands and therefore compensates for us killing the MIF5. So that told us that the redundancy isn't 2 genes in the same cell, but actually what we call separate cell lineages. And that's what this experiment shows, and I won't take you through that. We'll go on to the final story, and that is neuropsychiatric disorders. So the first thing you might ask is, why would you be studying behavior in a mouse? And particularly neuropsychiatric disorders, that is, human disorders, which we think ourselves as being much, much more complex than a mouse. And we got into this sort of fortuitously. These are a set of genes, and they're called Hox genes. And Hox genes are genes that turn genes on and off. Okay? And they're important for making sure that every part of your body is where it is. that I have my arms here, that my heart is here, my liver is here, my stomach is here, and so on. It makes sure that all the different parts of the body are made at the appropriate places and then connected together, and you have the appropriate plumbing. And it takes a lot of these genes and they work in combination, and that's why they can do a lot of different things. And each of these acts on 1,000 other genes, turning things on and turning things off. So it's quite complicated. And we've inactivated all of these genes and asked what happens in a mouse. And the gene that I'm going to be talking about is HOXB8. And we, in this case, is another graduate school student, and her name is Joy Greer in the laboratory. She inactivated that gene, and it was, she looked at the mouse, and the mouse looked pretty normal. So then she said, well, I'll look at its behavior. And what she did was to take movies. And mice do most of their activity at night, so you use an infrared camera. And she stood there 24 hours taking movies of this mouse, and then looked frame by frame to see what is this mouse doing. Okay? And everything was the same, except grooming. And this is showing the pattern of grooming. Okay? And all mammals groom, and we all do it for the same reason, and we do it with the same pattern. We go into the shower, we lather our hands, we start washing our head, we work down our body, and that's what's shown here. They wet their paws, they start working on their head, working on their body, and then finally end up in the tail. And that's true of all mammals. If there's anybody in here who doesn't have that activity, please see me afterwards. So this is actually some of the data. So these are SIP pairs, they're brothers and sisters. And black bar is that they have a mutation in HOXB8, and the gray bar is a normal, control normal. Okay, so in this case, she's looking at eating patterns. How much time do they spend eating? And then if you average it up, what you can see is the mutants of wild types are spending the same amount of time. And she looked at drinking, running around the cage, building nests, walking upside down in the cage, you name it. Whatever activity, everything was the same except for grooming. And that's shown in the next slide. And what you can see is that these mice spend twice as much time grooming as normal over a 24-hour period. The behavior of grooming is identical to the wild-type mouse. It's the same pattern. It's simply the number of grooming baths per 24 hours, and the duration of grooming is now longer. Okay, so they're spending about twice as much time. Now you can also induce grooming. You can spray a mouse with water and that starts to groom. Okay, and that's what we call induced grooming. And again, you can see the mutants spend twice as much time as grooming as the normals. Now I told you one slight error, and that is if we look at how much time do they spend sleeping. Okay, what you can see is that the mutants spend less time sleeping. Because we're looking over a 24-hour period. If they're spending twice as much time doing one thing, they have to be taking it away from something else, and what they take it away from is sleeping. So they're sleeping less, but grooming more. Now I'm going to show you that the grooming is pathological. Find my arrow. There we go. So what you'll see is when the mouse is grooming away, and then eventually it's going to turn. And what you'll see is when it turns, and these mice just continue to groom. When it turns, what you'll see is that they actually remove the body hair. There. Okay, so, and this is very similar to OCD. OCD, obsessive-compulsive disorder, what they do is, for example, wash their hands, continue to wash their hands until they have actually lacerations. Okay, and what we can see in these mice, that they're also pathological.